is get damaged. So the transport along the axon is very similar to the transport along the cell, where there's a cytoskeleton, which is served like a railway track, and there's cargo protein that carry a cargo. Here's cargo protein, carry a big cargo, and they will just walking along the track here to a certain direction. So those molecules and organelles are moved along those axons by the motor protein, which is this carrier, small carrier here, and also the element of the cytoskeleton, which is this big microtubule. By the direction how things are moved, we can just separate it to the anterograde and the retrograde. The anterograde send things to the axon terminal away from the cell body, something you made new, and send it to those direction, to those terminals. And those things include like a mitochondria and enzyme and also neurotransmitters, so things that your axon terminal will need. And the retrograde, like a retraction, so get everything back to the cell body. Remember, the cell body is the one who made all those make all those protein. So those are the retrograde. So if something get damaged or need to be recycled, you will bring it back to the cell body. In this picture, you see the neuron, which is yellow part. The branch part is dendroid. The big center is the body, cell body, and also there's a long axon. And along the axon, you see those bluish wrap on it. Those wrap are another cell, which protect your axon from damage, and also protect all those signal inside it, and also make the transduction of signal fast. So those wrap are called myelin. Myelin is a whitish protein wrap of your axon. So those myelin are in different segment, as you see in this picture, and we call it myelin sheath. And for the long axon and large diameter axon, they do have those myelins, but not everyone have it. If the very short ones or very thin axons, they normally do not have myelin. So the one have myelin, we call it myelinated fibers. Those wrap up those axons, which is shown in this picture here. The myelin is very important for the axon, so it protect and insulate the axon, and also very important thing is it increases the speed of the nerve impulse. So action potential can jump just like the kangaroo here, and jump, make those transmission faster. Okay, so for the non-myelinated fibers, they have to connect things continually, more like this. And they have to depolarize every inch of the cell membrane, so it's just kind of slow. Okay, so for the one with the myelin, just need to check. As long as those parts get a signal, and the signal will be conduct. So in the PNS, those myelination are by Schwann cell. Schwann cells are the one form those myelin sheaths here. Those Schwann cells, each one segment is one cell. And what happened is those Schwann cells wrap up around the axon in jelly roll fashion. Just wrap it up and push all those cytosome and cytoplasm to one direction. So what you get, those wrap here, like in this picture here, is a multi-layer of the cell membrane. Because the cell just wrap and wrap and wrap, what you get is a lot of layer of the cell membrane here. What is a cell membrane? What is a chemical composition of the cell membrane? Mostly it's a lipid, right? Lipid are here. Lipid can isolate and insulate those axons to prevent the electrical signal to go out of the axon there. Okay, so that is a concentric layers made by the Schwann cell membrane, and that is called a myelin sheath. Okay, so the layers of different of those cell membrane. Okay, that's a myelin sheath, and this picture here, what you are looking at is a cross section of axon. So you see those Schwann cell wrap it up. When the Schwann cell try to wrap up an axon, what happened at the beginning is it just envelope an axon like in this picture showed, and then it just a coil on it. So rotate around the axon, wrap it up. At this time, it's still wrapped up loosely. And then those cytoplasm is kind of forced out, so you see the myelin sheath is formed. So those cytoplasm are pushed out to the outermost layer. So that's why when you see those myelin sheaths in this picture here, the middle dark part, those dark part here, those are the cell membrane. Those are the many layers of the cell membrane, just like this part. And in fact, you do have the out layer, which have a little bit cell, the cytoplasm, and also the nucleus. Don't forget, Schwann cell is a cell, so the nucleus are there. So the outmost layer here, you see there's a lot of cytosome there. It's kind of transparent compared to the myelin sheath. That is the part that contain the cytoplasm nucleus of the Schwann cell. Because the outermost layer have those nucleus and all those other important functions unit of the Schwann cell, so that part we call the outer collar. It's outer collar of the Schwann cell or perinuclear cytoplasm. And you can see here's a nucleus and there's a nucleus. So those part is the out layer of the Schwann cell, which contain the thing that will maintain and keep the Schwann cell alive. And inside the myelin sheath, for the myelin sheath which is so dense, which is this part, so those parts have no cytoplasm, very little proteins, no channels, no carrier. So basically, those are just a phospholipid. So it's a very good electrical insulator and prevent those electrical signal to go out. So they do have some interlocking protein here. Interlocking protein is like the adhesion, it just connect and bind all those myelin sheaths together to make sure it's close and also fixed at that location. As you can see here, the myelin are in segments. So there's a different segments, and between those segments, the gap are called the node of Ravier. The node of Ravier are the gap between the myelin sheaths, which is a gap, and those are the place that allow those axon to check the signal to recharge. Every time the axon have to recharge their signal at those node of Ravier there. And for the axon that do not have a myelin, called non-myelinated fibers, they are normally very thin and short because the signals cannot transfer so very far and it's too slow to send to faraway location. Those fibers are not wrapped in myelin. It do have some Schwann cell surrounded, but not form a myelin sheath like the one we just saw. And in the CNS, it used the oligodendrocyte to form the myelin sheath. It's not a Schwann cell here, it's oligodendrocyte. Oligodendrocyte have a branch, and one oligodendrocyte can form a myelin sheath for different axons, like shown in this picture here. It just extend a branch, and this branch will wrap up an axon. So the cell body are in the middle, and it doesn't really have the collar part as a Schwann cell have. Okay, so for one oligodendrocyte, it can wrap up to like a 60 axon at once. But however, for one Schwann cell just wrap up one axon. So for those CNS, For CNS, there's a node of a Ranvier are present as well. There's no outer color because the cell membrane, the cell nucleus, have their own cell body outside. The thinnest fiber are not wrapped, not myelinated either in the CNS. Those can be covered by nearby neuroglia, and those normally the signal cannot transfer or transmit too fast as well. In the CNS, normally if you see a section of the brain like this picture showed, you see the gray outside and white inside. So the white matter inside are normally a region of the brain that have a lot of myelin sheaths. So myelinated fiber are there, that's why make it white. And for the gray matters, cell bodies are on gray matters, and also the non-myelinated fiber are in the gray matter as well. Okay, at the end, let's see a few review questions here. Nerves are found in where, and while the tracts are found in? The answer is A. Nerves are in the PNS and tracts are the nerve fibers in the CNS. Next one. The portion of an axon that communicates with its target cell is? The answer is axon terminal. So the portion of the axon will send information to the target cell is at the axon terminal. That's the junction, that is a connection between a neuron and a target cell. Last question. The basis of a differentiation between gray matter and white matter in the CNS is the presence of what or in the white matter? The answer is myelinated fiber, because it's myelinated and it makes it white.